All right, let's get straight into it. We are diving into a massively high-yield explainer on the cardiovascular system today. If you're prepping to absolutely crush the MCAT, this is exactly what you need. We're not just going to passively review facts. We're going to master the underlying concepts and those crucial structure-function relationships that test makers love to go after.
Here is our game plan. We're going to cover anatomy and blood flow, conduction and the cardiac cycle, blood vessels and circulation, blood components and types, cardiovascular physiology, and finally, gas transport and coagulation.
Let's start with Section 1, Anatomy and Blood Flow, focusing on the heart as a pump.
Now, it's a really common misconception that the heart is just one single, simple pump. It's actually two distinct pumps operating in series. The right side of the heart handles deoxygenated blood, pushing it right next door to the lungs. That is your pulmonary circulation. Meanwhile, the left side of the heart receives that newly oxygenated blood and pumps it out to the rest of the body, which is your systemic circulation. Before blood can leave the heart, it moves from the atria into the ventricles, passing through the atrioventricular valves. You do not want to fall for the classic test day trap of mixing up which valve is where, so just remember this incredibly useful mnemonic, lab rat. Left atrium equals the bicuspid or mitral valve, right atrium equals the tricuspid valve. These valves are absolutely vital because they prevent blood from flowing backward when those powerful ventricles contract.
Tracing the complete circuit, blood comes in from the superior and inferior vena cava, moves through the right heart, out to the lungs, back into the left heart, and finally gets forcefully ejected through the aorta. But as you trace this pathway in your head, never forget the cardinal rule of blood vessels. Arteries always carry blood away from the heart, and veins carry blood toward the heart. This rule is absolute, regardless of oxygenation. That's exactly why the pulmonary artery carries deoxygenated blood, while the pulmonary vein carries oxygenated blood.
Okay, so here is a huge structure-function takeaway. There is an immense difference in muscle mass between the two ventricles. Think about it. The right ventricle only has to push blood into a low-resistance circuit right next door to the lungs. But the left ventricle, it has to be far more muscular because it's overcoming massive systemic resistance to pump blood all the way up to your brain and all the way down to your toes.
Section 2, Conduction and the cardiac cycle. Let's shift from the physical pump over to the electrical wiring that drives it.
Unlike skeletal muscle, cardiac muscle actually has myogenic activity. That means it generates its own beat, entirely independent of the brain. The signal spreads super fast via intercalated discs and gap junctions. Now, the autonomic nervous system, like parasympathetic vagal stimulation, can slow the rate down, but the impulse itself starts at the SA node, pacing you at about 60 to 100 beats per minute. From there, it hits the AV node. And here's a massive high-yield detail. There is a critical, built-in delay at the AV node. Why? So the ventricles have enough time to completely fill with blood before they squeeze. After that brief delay, the signal rushes down the bundle of His and spreads right through the Purkinje fibers.
The cardiac cycle can seem notoriously complex, but let's simplify it by just thinking about the physical sounds of a heartbeat, that classic lub-dub you hear through a stethoscope. The first sound, S1, or the lub, happens right at the start of systole when the AV valves snap shut to stop blood from shooting back into the atria. The second sound, S2, or the dub, happens at the end of systole when the semilunar valves snap shut to prevent backflow into the ventricles. If those valves get damaged or don't close perfectly, you get turbulent flow, and that's exactly what you're hearing when someone has a heart murmur.
You also have to be able to map that physical pumping cycle onto an EKG. You need to know exactly which electrical event corresponds to which wave. The small p wave, that's atrial depolarization. The massive QRS complex, that is ventricular depolarization, which makes perfect sense given how much thick muscle the ventricles have. Finally, the T wave is ventricular repolarization, which just resets the whole system for the next beat.
Here is an absolute must-know formula. Cardiac output equals heart rate times stroke volume. It's simply a way to calculate the total volume of blood pumped per minute. If your heart rate is 70 beats per minute and your stroke volume is 70 milliliters, your cardiac output is going to be roughly 5 liters a minute. But watch out for the MCAT application here. You'd naturally think a faster heart rate always means a higher cardiac output. But at an extremely rapid heart rate, your cardiac output can actually plummet. Why? Because the heart is beating so fast that the ventricles literally don't have enough time to fill up with blood during diastole.
Moving to Section 3, Blood vessels and circulation. Let's follow that cardiac output out of the heart and check out the vascular plumbing.
Comparing the different vessels is a classic test topic. Arteries are thick and highly muscular. Once they fill up with blood, they use this incredible elastic recoil to maintain high pressure and just force that blood forward.
Veins, on the other hand, are thin-walled and very compliant. They actually act as storage tanks, holding about 3/4 of our entire blood volume. And because veins have to fight gravity to get blood from your legs all the way back up to your heart, they rely heavily on one-way valves and the squeezing action of your surrounding skeletal muscles. If those valves fail, you end up with varicose veins. And if blood just sits there and stagnates, it can form a clot, like a deep vein thrombosis, which can break off, travel, and cause a deadly pulmonary embolism.
Continuing with our plumbing system, it is incredibly high yield to know that arterioles, which are the smaller muscular arteries, create the single greatest drop in blood pressure in the entire circulatory system. From there, the blood hits the capillaries. Capillaries are so thin, literally just one single endothelial cell thick, allowing for perfect gas and nutrient exchange. Also, usually blood flows through just one capillary bed and heads straight back to the heart. But there are exactly 3 portal systems in the body, the hepatic, the hypophyseal, and the renal, where blood uniquely passes through 2 capillary beds in series before heading back.
Section 4. Blood components and types. Now that we understand the pump and the pipes, let's look at the actual fluid flowing through them. Blood is basically a mix of plasma, which is the watery solution of nutrients, proteins, and hormones, and the formed elements, which are your erythrocytes, leukocytes, and platelets. The percentage of your blood that is red blood cells is called your hematocrit, and their production gets stimulated by erythropoietin from the kidneys. Now here is a massive trap. To make maximum room for carrying oxygen, mature red blood cells actually spit out their nuclei and their mitochondria. Think about what that actually means. Without mitochondria, they cannot perform aerobic respiration. They have to rely entirely on glycolysis for ATP. This is a brilliant evolutionary design so they don't consume the very oxygen they are trying to transport.
For blood types, you need to understand that AB alleles are co-dominant. O negative is the universal donor because it lacks A, B, and Rh antigens, so it won't trigger an immune response in anyone. AB positive is the universal recipient. You absolutely must understand the Rh factor as well, which is a dominant trait. If an Rh negative mother carries an Rh positive fetus, her body might create antibodies against that fetal blood. In a future pregnancy, those antibodies can actually cross the placenta and destroy the new fetus's red blood cells, which is a fatal condition called erythroblastosis fetalis. This is exactly why doctors administer RhoGAM, to prevent that maternal sensitization from happening in the first place.
Section 5. Cardiovascular Physiology. Let's apply some basic physics to how this all works.
How exactly does the body maintain blood pressure? First, know this equation inside and out. Delta P equals cardiac output times total peripheral resistance. This is literally just Ohm's law applied to the human body. The body controls resistance through vasoconstriction and vasodilation, and tightly regulates fluid volume using baroreceptors that detect vessel stretch. If your pressure drops, hormones like aldosterone and ADH kick into gear to retain water, raising both volume and pressure. If pressure gets dangerously high, the heart itself releases atrial natriuretic peptide, or ANP, to promote salt and water excretion and bring that pressure back down.
Down at the microscopic capillary walls, fluid exchange is a constant physical tug-of-war dictated by Starling forces. On one side, you have hydrostatic pressure, generated by the pumping of the heart. This acts like a leaky hose, forcefully pushing fluid out of the capillary. On the other side, you have plasma-oncotic pressure, generated by large plasma proteins. This acts like a sponge, osmotically pulling fluid back into the capillary. Any excess fluid left behind in the tissues just gets swept up by the lymphatic system and returned to circulation.
Section 6. Gas transport and coagulation. Let's zoom all the way down to the biochemical level to see how we exchange gas and repair the vessels.
The hemoglobin-oxygen dissociation curve has a very distinct sigmoidal or S shape. That shape proves that hemoglobin binds oxygen cooperatively, meaning the more oxygen it binds, the easier it becomes to bind the very next one. You absolutely have to know the Bohr effect. When you exercise, you produce more CO2, more hydrogen ions, and more heat. All of these factors cause the curve to shift to the right. A rightward shift means hemoglobin has a lower affinity for oxygen, meaning it'll drop off more of its oxygen payloads exactly where those starving, exercising tissues need it most. Also, remember that increased 2,3-BPG causes a right shift too, whereas fetal hemoglobin is shifted to the left, which gives it a higher affinity to pull oxygen away from the mother's blood.
Zooming in even further, we find the bicarbonate buffer system, catalyzed by carbonic anhydrase.
Carbon dioxide is primarily transported in your blood not as a gas, but as this soluble bicarbonate ion. The specific equation explains exactly why an increase in blood CO2, like if you were to just hold your breath, drives the reaction to the right. This leads directly to an increase in protons, and therefore a dangerous drop in your overall blood pH.
Let's wrap up by talking about how the body patches up the pipes. While doctors in the 19th century used to apply 40 million leeches a year to stop blood from clotting, our bodies actually rely on a rapid coagulation cascade the second a vessel is injured. It kicks off with a platelet plug, followed by a complex cascade of clotting factors. Ultimately, the enzyme prothrombin is converted into active thrombin, which then converts soluble fibrinogen into a really tough, stable fibrin clot that nets everything together like a microscopic web. We know that an enzyme called plasmin eventually breaks this web down once it's healed. But consider this. If this delicate, highly complex cascade of prothrombin and fibrin is thrown off by even a single genetic mutation, your blood either won't clot at all or it'll clot unstoppably inside your own vessels. How does modern medicine intervene in this cascade to save a life when the system fails? Well, that is a biochemical puzzle for you to solve next. Keep questioning the whys, and we will see you in the next explainer.